Okay, good afternoon, 10 Hubble. Welcome to, I think now, episode 10 of Mr. MALTBY'S Home and Learning series. This week we're looking at the rate of decomposition, linking into our cycles that we've been looking at over the last couple of weeks. So let's get straight into this. We're going to be thinking about the various conditions needed for decay, and then we're also going to be doing some calculations to look at the rate of decay, rate of decomposition. So state the two types of organisms involved in the process of decay. Just to get us thinking. In terms of deeper thinking, suggest what affects the rate at which dead animals and plants decay. Okay. Now, you can pause the video and have a little think about those. And I'm just going to say I've got a time lapse here, okay? An interesting time lapse of this rabbit. This is a rabbit that has died, natural causes. And I'm just going to show you this time lapse. It's really interesting. Now, word of warning, if you are a little bit squeamish, then you might want to just skip ahead about just over a minute, okay? Just over a minute if you don't want to see this time lapse of this rabbit decaying. But those of you that are interested, it's really interesting to see how rapidly, okay, this rabbit does decay and break down. So if you're a little bit squeamish and or you are particularly fond of rabbits, then you want to move ahead about a minute. If you're interested, you can stick to this and think about what's going on relating to these couple of questions that we've got going on. So let's have a go. Okay, so obviously this is a time-lapse video. You can see in the top right-hand corner of the time-lapse, you can see we're on day 2 now. Okay, you can see the insects. You obviously won't be able to see the microorganisms, but you can see how rapidly this animal is decaying. And of course, yes, this might seem a little bit gross to some of you. You can see there, you can see the flies, you can see the rabbit decaying rapidly over this time. But of course, and this is really, really important to consider, that decay and animals and plants decaying, dying and decaying, being broken down, is necessary for life. Okay? And that's really, really interesting. Okay, I'm going to explore that idea. Into day 7, you can see now how much it's decayed down. Okay, so what we need to consider today is how that process happens. What are the conditions needed for decay? Okay, that's what we're going to be exploring. Two types of organisms. Well, we've got microorganisms such as bacteria. Okay, we also have various other microorganisms that are going to be involved in decay, as well as what we refer to as detritivores, organisms that feed on detritus, dead materials such as maggots, worms, and other insects we refer to as detritivores. So the two types of organisms we've got, microorganisms and detritivores. Suggest what affects the rate at which dead animals and plants decay. Well, you might have some ideas there. I'm not going to say just yet. You might have some ideas about what is needed. Thinking about, really, if we've got these microorganisms and detritivores, what do they need to survive in order to feed and decay dead animals and plants? And that's what we need to consider. Okay, so thinking about the focus for today, we've got our keywords here: temperature, moisture, oxygen, decay, bacteria, and fungi. We've also got these learning outcomes here, so apply factors which affect the rate of decay to real life situations. For example, compost making, with some questions around that, preserving food as well. I recently posted on Teams a really interesting BBC documentary all about decay. It is fascinating. It's probably one of my favourite documentaries that often we show at school, and definitely worth having a look. I think it's just over an hour and a bit, hour and a half maybe, but it's really, really interesting. And one of the videos actually looks at how the US Army uses the knowledge around decay to preserve food, okay, and food supplies for the army, which is obviously really, really important. Explain why factors speed up or slow down decay, and list the factors that affect the rate of decay. So some really interesting stuff. Those of you that are interested here, decomposition then is specifically biology only, so we are doing triple content here. and you can look at the specification here in terms of what we need to be able to do. And we are, of course, going to calculate rates and changes here. So the conditions for decay. Okay, if you want to make some notes on this, some additional notes, that's up to you, or if you just want to follow along from the video, that's also absolutely fine. So decomposition is vital for the recycling of materials in an ecosystem. It is vital for life. Factors which affect the rate of decay. So temperature. Chemical reactions in microorganisms work fast in warmer conditions. Too hot, it stops. Too hot, if the temperature is too high, those reactions will stop because the enzymes in the decomposers will denature, and decay slows down or stops if conditions are too cold. We know that chemical reactions work best at an optimal temperature. The same applies for these microorganisms. Optimal temperatures are key, and if they are too high, we know that these enzymes will denature. Too low, and it just decreases the rate of these reactions. We can relate that to a chemistry topic. Moisture prevents microorganisms drying out, and most grow faster in these moist conditions. So they prefer the moist conditions for their growth. If there's more microorganisms, that means more microorganisms for decay. And then oxygen. Most decomposers, referring to both our microorganisms and detritivores, will respire aerobically, as in they need oxygen. So they need oxygen to break down food and to transfer energy to grow and reproduce. Decay occurs rapidly in these aerobic conditions. If we have these microorganisms, they are living, they need to respire in order to live, to reproduce, and therefore allow decay to happen. If we have oxygen, that allows the decomposers to respire, it allows them to break down those materials in respiration to release energy, also to survive and to grow, to reproduce. More oxygen means more decomposers, which means more decay. So these are three factors: temperature, moisture, and oxygen. And I guess we can, think about the flips. An interesting thing to do right now is to think about the flip side of that. Okay, so if we want to stop things from decomposing, think about our fridge and our freezers, for example. We have a really, really low temperature. We will ideally reduce the moisture. We will put them in sealed bags, or we will put food in Tupperware boxes so there's no oxygen going into those bags or boxes. So if we think about how we preserve food, we do the opposite. We remove these factors to stop our food from decaying. Of course, if we buy an apple or whatever the food is, there will be some bacteria, there will be some microorganisms on the surface of those products. So by having a low temperature in our fridge, by taking away the moisture, and by removing the oxygen and sealing them in bags and boxes, we aim to stop the decay process of our food. That's an interesting way to think about it, to think about the opposite. How do we preserve food? Okay, now on Teams, I sent you a worksheet. This is an optional worksheet, okay? And you can go through this worksheet digitally. You can pause the video at various points to consider these questions, or if you're able to have the worksheet in front of you, you can pause the video now, have a go at the worksheet, and then go through it. So there's a couple of options here. So to get us thinking deeper, if we're turning over the contents of a compost bin every so often, can increase the rate at which decomposition takes place. Explain why. So pause the video, have a think. Why is it that we turn over, our compost heap? Now, one of the main reasons really is to aerate. To aerate that compost heap, to increase the levels of oxygen by turning it over, getting oxygen back into that. That relates directly to our factor that is needed for decomposition, which is oxygen. Oxygen is needed for our microorganisms to respire, to therefore respire, release energy, reproduce, grow. More microorganisms means more decay, and we need to provide that oxygen to allow those microorganisms to survive and reproduce. Another factor we may link this to is moisture. If we have a compost heap, the top may dry out. If we turn it over with redistributing that moisture, that could also help with the decomposition process. Okay, so into some of these questions then. Again, this is the worksheet that was sent to you. Choose from these pairs of conditions the one which would speed up rotting. So it's fairly straightforward just to get us eased into this. So of course, warm conditions, air of course contains oxygen. We need oxygen for the decomposers. Wet conditions and stirred. By that we mean it's turning over our compost heap. So what is compost? Compost is decomposed garden waste. Why is it useful to gardeners? Well, compost is useful as a natural fertiliser for plant growth, except soil conditioner, lower soil pH. So it's really useful as a natural fertiliser because it contains lots of nutrients. Because things have been broken down, which then makes it available to plants. 2 gases can be given off during decomposition. Write a short passage about why each one is important. Well, carbon dioxide is a greenhouse gas, but also as a reactant in photosynthesis. And methane is a greenhouse gas, but also as a biofuel, which we'll come to later. So coming back to this question then, I did say it verbally, but we've got the answer here then as well. So many microorganisms are involved in decomposition and need oxygen for aerobic respiration. Turning over compost bin contents increases air mixture, and turning compost bin contents increases the rate of decomposition as microorganisms have plenty of oxygen for aerobic respiration. So it's tying in to oxygen and aerobic respiration that the decomposers need. Okay, onto our biogas generator. It's really interesting here. So at this point it might be good to pause the video, just double check your answers for those questions before we move on to this section. So when bacteria decompose waste material in anaerobic, that's without oxygen conditions, they produce methane, okay, methane gas. Methane is a flammable gas and can be used for biofuel and therefore for heating, cooking, generating electricity, fuel for cars. And to do the above, you need a biogas generator. Okay, so plant and animal waste is put into a biogas generator. Anaerobic conditions are created as a result, and the bacteria produce methane. So when we get decomposition in an anaerobic condition, we're able to produce methane, and that methane then we can use for various applications. Okay, really interesting stuff. Bacteria work best at about 30 degrees, and biogas generators must create optimum conditions to be effective. Biogas generators are a clean, renewable energy source. Okay, so if you didn't get any notes about biogas generators from the textbook, let's make sure we've got these into our exercise books, please. Okay, we've got a short little video here for you to watch.
The theory is simple, but I've got no idea how to make this work properly, so I'm off to North Wales on a fact-finding mission. I've heard about this man who extracts methane from poo, which you can burn. I don't know how he does it, I don't know the technique, I'm very interested to learn. What I do know is, however, he doesn't use human poo, he uses dog poo. This experimental poo power scheme is the brainchild of former banker Gary Downey. This is one of your stations?
Yes, that's the dog station. We're going to collect the dog waste from here and we're going to turn it into energy.
How much dog poo is there? I mean, not here, but I mean across the whole country.
Well, in the UK wide, we reckon that there are between 1,000 to 1,200 tonnes of dog waste created every day.
How much methane could you get out of 1,200 tonnes?
Quite a lot. One average dog sitting equals the electrical energy to make 2 cups of tea or coffee.
That is astonishing. I'm interested to find out how this works, but first we need some raw material. Oh, that's disgusting. That's wet, that one. Okay, I'm going in. I know from experience, it's bad enough. When it's somebody else's dog, it's like picking up somebody else's poo. Has he done one yet?
Yes, he did one over there.
Oh, I think I've already got him. 1,200 tonnes of poo from dogs a day in Britain. That would power an awful lot of stoves. Ah, that's my kind of poo. That is my kind of poo. I call them soft stools. That was a joy to pick up. I'm interested to see how much gas you can get out of, you know, a bag full of poo. How much energy is in this? The answer lies down the road, where I'm taking my haul of doggy doos to be processed at the poo power station. Whoa! No, that's not it, is it? My godfathers. If that tank is full of poo, making biogas, then they're producing it on a sort of NASA scale here. This place already transforms cow dung into biogas on an industrial scale. But they've recently started to explore the potential of dog muck as a source of free energy.
Aha! The nerve centre of poo.
Engineer Jonathan Tomlinson is the owner.
So here, in that big tank, you are digesting poo.
Yeah, cow muck just goes in that.
Are you then turning that into electricity?
At the moment, we've got one 80 kilowatt generator running flat out.
And that would be enough to power a large housing estate.
Well, enough to power 160 houses.
Yeah,
which is
exactly.
A small village.
John's system is the most advanced in the UK. Now I'm going to see for myself how it's done using the experimental dog poo digester.
That's my collection. That's my little bag from this morning. I was hoping we might just kind of be able to somehow cleanly just drop the bag in.
No, I'm afraid not.
No, you have to empty it all out.
Yeah, well, that's be done manually. We've got to get it into a paste so we can pour it in. So as soon as it goes in, it starts to digest.
A paste. I know what's coming. Mmm. That's not a nice one.
That's the consistency you want. So that's the good stuff. We walk over to the tap. So about 3/4 full. Start squelching.
Do you know what? I can do this, I just can't look at it.
Once in the system, bacteria will break down my poo paste in a process called anaerobic digestion.
That's bubbling. You can hear it. Fart gas.
We can boil some water.
Make some tea, some dog poo tea. Whoa! Look at that. What a perfect flame.
What I come away with is this overbearing sense of how incredibly wasteful we are and how incredibly inefficient we are at managing the resources that we've got.
Shall I be mother?
Please be mother. That's a good rolling boil, as they say, which is what I like to see. What's that with?
Is there a Thai poo joke coming up now?
So much easier, isn't it, just to go and buy another can of propane. So much easier to install another gas pipeline across Russia. And yet on our doorstep is all this stuff we could use. What's wrong with us?
Okay, really interesting video there. And this image here is kind of just explaining that process. Okay, so we've got our waste. Okay, the waste goes into our generator with our bacteria respiring anaerobically, decomposes that waste, releasing our methane gas. That methane gas is then collected, and that gas can then be used for various applications like cooking and heating. But of course, in burning that gas, we will of course be releasing carbon dioxide back into the atmosphere. But then of course, we've got a nice link there actually to our carbon cycle, as much as we've got combustion here of that gas, okay, releasing the CO2 in the atmosphere, and then the plants are then able to use that CO2 in photosynthesis. So this is a quite nice little diagram that links over. Yes, we've got a renewable energy source here, but that links over also to our carbon cycle. So we want to draw this as an example into our books. So pause the video, have a go. Okay, so on to calculating the rate of decay, and you should have also got another worksheet for this, which is available. Again, we're going to go through it digitally here as well. So we're going to look at how we calculate the rate of decay. Okay. A group of students investigate the decay of bread in the lab. They find that in the first 5 days, the bread had decreased in mass by 2.5 grams. So we're looking at mass. How does the mass change? In the next 5 days, it has decreased by a further 5 grams. Compare the rate of decay of bread between the first 5 days and the last 5 days. Okay, so quite a complicated step. Okay, so first of all, we're going to write down the following formula. Our rate of decay is the change in mass, okay, in grams divided by time. Okay, in this case, we're looking at grams and days. Okay, because it's asked specifically for that. You may need to change units. Okay, but the questions are specifically for that. Fill in the values. Okay, so we've got fill in the values for the first 5 days, which is our rate of decay, 2.5 grams over 5 days, and calculate the rate over the first 5 days, which is our rate of decay, which gives 0.5 grams per day. It's the slash there, meaning per day. Finally, in step 4, fill in the values for the last 5 days. So our rate of decay is 5 grams over 5 days. Calculate the rate of the decay over 5 days, which is a gram per day, and then we compare the two rates. So we've got 0.5 per day compared to a gram per day, and then we can compare those two rates. The rate for the last 5 days is 2 times the rate of that for the first 5 days. Okay, because 1 gram per day is obviously twice as much as 0.5 grams per day. Okay, and there's our answer. So as long as we've got our formula there at the top, we can work through this step by step. And if you want to, you can write this down as an example. Maybe take some time now to pause the video and go back through this and make sure that you understand this. So onto our worksheet then. We've got some tasks here to do. And thinking about these questions here. Okay, so you want to pause the video and just have a go at these questions, and we will go through the answers. Read the questions carefully, consider what is actually being asked, and remember the steps, okay, to show your working. It's really important that you show your workings here. So I'll read out the question, we'll go for the answer shortly. Elliot was investigating how the volume of water available affected the rate of decomposition of a piece of bread. He placed pieces of bread in dishes and added various volumes of water. The bread was then left for 20 days. State what type of microorganisms mold is. Interesting. Compare the rate of decay of the bread with 0 milliliters of water added to the bread with 3 milliliters of water. Suggest reasons for these different results. So lots to these questions, lots of aspects of this question. Break it down, work through it methodically. We'll look at the answers shortly. So here are the questions then from the worksheet that was also posted on Teams. Again, optional questions here, which we'll go through now. If you want to pause the video first and have a go at these questions now, you can do that now. So pause the video and have a go. And then we'll go through the answers. So first question. Calculate the rate of decay of a piece of fruit that reduced in mass by 70 grams over 28 days using the formula. Okay, so rate of decay is grams per day. So change in mass over time. Here then we've got 2.5 grams per day. Okay, simple, straightforward calculation there. Change in mass. Okay, it reduced its mass by 70 grams. That's our change in mass over time, which is our 28 days. A group of students were studying the rate of decay of a piece of bread. At the start of the experiment, the mass was 5 grams, and after 10 days, the mass of the bread was 20 grams. So automatically, we can underline those values. Remember, good exam technique, and we can see that the change in mass will be 5 grams. So calculate the change in mass of the bread over 10 days, 5 grams. Calculate the rate of decay of the bread over 10 days. That's straightforward. 5 over 10 is going to give us our 0.5 grams per day. A banana decayed by 0.1 gram per day in the first week, and in the second week it decayed by 1 gram per day. Compare the rate of decay between each week 1 and week 2. So first week we've got this 0.014, second week 0.14. The banana decayed 10 times faster in the second week, so 10 times faster. Okay, Molly investigated the decay of oranges in the lab. She found that in the first 2 weeks, the oranges had decreased in mass by 5.6 grams. In the next 2 weeks, they had decreased by a further 10.5 grams. A further 10.5 grams. Calculate the rate of decay of the oranges in the first 2 weeks. 0.4 grams per day. Okay, Again, using that calculation, fairly straightforward. Calculate the rate of decay of the oranges in the second 2 weeks. Again, using the equation and finding a difference, we've got our 0.75 grams per day. Compare the rate of decay of the oranges between the first 2 weeks and the last 2 weeks. Again, the rate of decay increased by 88% between the first 2 weeks and the second 2 weeks. So a good opportunity here to show it as a percentage. Okay, this is the extension question. You've got the answers here. I'll just pause the video now and you can have a look at these answers. For those of you that had a go at the extension questions, you can have a go at this now. So you can pause the video, okay, and then you compare your answers to what you have got. Okay, and last little video just to round things off.
Think of all the food made in the world each year. Hard to picture? Then imagine that you are all of humanity, and on a plate in front of you is the one lovely annual meal you make for yourself. You did all sorts of work putting that meal on your table. You must be eager to consume the fruits of your labor. And the vegetables and meats and waffles of your labor too, right? Well, oddly enough, a third of that meal ends up in the trash. A third of the food we eat globally. An estimated 1.3 billion tons ends up as waste. All the work we put into producing that food is wasted. And what's worse, it costs us. America alone spends an estimated $165 billion a year managing food waste. We're wasting food, energy, and money. Perhaps worst of all, we're wasting the chance to change, to make the system of food consumption more efficient. If you want to bring on that change, you should know about a humble yet diligent and ever so crucial ally, the worm. Worms convert organic waste and other compostable products into natural fertilizers. Up to 75% of what we put in the waste stream can become food and bedding material for vermicomposting. You can create a worm bin in your own home to see the composting process in action. First off, you need worms, and not your typical earthworms. You need red worms, Eisenia foetida, the species responsible for most vermicomposting in North America. These red wigglers are surface dwellers who don't burrow too deep. They're optimal feeders around room temperature, and they're well suited to converting organic waste into usable fertilizer. Now, your worms might be vermin, but they need a comfortable space to live and work. Some bedding material, either shredded paper or cardboard, some moisture, and of course, food. Namely, your leftovers, slightly decomposed table scraps. The worms break down food waste and other organic matter into castings, a fancy synonym for worm poop. Their excrement is absolutely teeming with microbes, which continue the decomposition process, making all those once wasted nutrients available again as fertilizer. The timeline for the whole process varies depending on the quantity of worms, the temperature, and how much waste is added to the bin. And there's another timeline to consider. In a healthy worm bin habitat, worm reproduction will occur when the wigglers become sexually mature, indicated by an elongation of the segments into a bulbous structure. 3-month-old wigglers can produce 2 to 3 semi-translucent yellow worm cocoons a week. You thought only moths and butterflies come out of cocoons? Well, we can't all be majestic. It takes around 11 weeks for new babies to hatch. When your bin seems to be full of living vermicelli noodles, it's time to share the bounty with your friends and start a vermicompost club. Or keep those worms to yourself and start a business. Vermicomposting isn't confined only to small worm bins. It's an emerging entrepreneurial enterprise. Large-scale facilities convert bulk organic waste and even manure into rich black castings called black gold. Its value as a soil additive is unparalleled, and it can help plants resist harmful pathogens. The lack of available land in urban environments, coupled with growing interest in smaller-scale farming, means there's a market for vermicomposting. Many communities use composting as part of zero-waste strategies, and they can sell their worm-eaten table scraps to local farms hungry for rich fertilizer. So instead of wasting money dumping wasted food in landfills, we can remake waste into an asset, putting it back into our food system to make it more sustainable. All with the help of the humble worm, the tiny organism that can help us change the way we look at food's place in our lives, and our place in the world, as long as we give the little guy a place at our table. Well, not an actual seat at the table. A bin in the shed is fine.
Okay, really interesting video there, which I thought would be a really nice way to finish this topic here. But I'm not saying that everyone should go out now and make their own compost heaps and worm bins, but food wastage has certainly been something I've been thinking about as we've been in lockdown. Certainly, perhaps buying too much food and some of it going in the waste. And I think this is a really nice way just to finish this topic for us to think about food waste. Okay, and that's something that's really important. Okay, for our world. So that's that for this topic. You want to pause the video now, go back through the questions and things, and make sure you've got everything spot on. And remember, I'm on Teams if you've got any questions. Okay, so thanks very much.